Welcome to Biology My Passion. I am Soumya Harikrishna. We will learn the chapter Microbes in Human Welfare. This chapter is comparatively simple, but you can expect a lot of questions, and there are a few concepts which are very important. So we know that the microbes are the small organisms which cannot be seen with our naked eyes. When we talk about their presence, they are almost found everywhere. You can see in these pictures, there are a few regions where no other organism can survive. For example, in the first picture you are seeing is the soil. They can live in extreme situations or climatic conditions as well. In the soil, these microbes are found in plenty. When it comes to several meter thick snow, because of the cold climatic condition, only certain organisms or animals adapted to that condition can live there. But there also we can find certain kind of bacteria. Or the regions where the acidic property or acidity is very high, so that it cannot support any life. But still we can find certain bacteria there. And hot thermal vents, where the average temperature is up to 100 degrees Celsius. There also no other organism can survive, but bacteria can. So from this we can understand that they are present almost everywhere. What are the organisms coming under the category of microbes? We know protozoans, bacteria, fungi, and microscopic plants, viruses, viroids, and also prions. Of this, protozoans are unicellular and eukaryotic organisms like amoeba, paramecium, etc. Whereas bacteria are very familiar to us. We have learned this as an example of prokaryotic organisms. Fungi, in that certain fungi are very small, like their hyphae are small, and also some microscopic plants. Viruses also we are familiar with. But what are these viroids and prions? Viruses don't have a cell. Virus has only a protein code with a genetic material. It can be either DNA or RNA. But sometimes it's all the more interesting. The only genetic material will be there without a protein code. Then we call it as viroid. The next is again interesting. Only protein. There is no genetic material also. It's a highly infectious agent, and that is called a prions. We can culture several microbes in our lab in the artificial conditions. In the first picture you can see a petri dish with the agar as a base. In that you can see several bacterial colony. Each colony or the circle that you are seeing has millions of bacteria in it. The same way, the next picture shows some fungal growth, or they are otherwise called a mold. Whenever we hear about microbes, we think about all the harmful effects they are having on our life. For example, they cause various diseases to plants and animals, and also they cause destruction to many food items. They spoil a lot of food items. They can spoil leather and other goods. But at the same time, they have got some advantages as well. In this chapter, we are trying to forget about the negative sides of all the microbes, and we are focusing on the positive sides and how are they helpful in our daily life. All these advantages or the uses of microbes we are classifying or studying under these headings. First, how are they useful in household products? Then, how are they useful in making industrial products? Then in sewage treatment, that is wastewater treatment, production of biogas, as biocontrol agents in farming, and also as biofertilizers. Let's see one by one. Once we finish this, chapter is done. So first is household. Microbes in household products. The first thing is production of curd from milk. Microorganisms such as lactobacillus and other commonly called lactic acid bacteria or lab, they grow in milk and convert it into curd. During their growth, lab produce acid that coagulate and partially digest the milk proteins. So we know how to make curd at home. We boil the milk, bring it to room temperature or lukewarm condition. To that we add an already prepared spoon of curd. That is acting as a starter or inoculum which has millions of bacteria in it, so that the process becomes faster. The process of formation of curd is taking several hours. Usually we keep it overnight. The next morning the curd will be ready. So what are the advantages of curd over milk? Which is better, drinking as milk or curdling it? Actually, curd has got certain advantages than milk. First is vitamin B12 content in the curd is higher than that in the milk. And also it checks the harmful bacteria in our digestive system. Our gut has very numerous bacteria which are helping in our digestion. But at the same time, there can be harmful bacteria that is coming in through our food also. So these bacteria are controlled by the good bacteria, that is lactic acid bacteria present in the curd. That's why if you see any traditional cuisines, curd or in different forms, it can be in the form of laban or raita or salad, it is a part of any traditional feast. For example, if you take biryani, you take salad with it. If it is fried rice, you can take raita with it. Or if it is a traditional feast in South India with plantain leaf also, at the end laban will be served. This is for increasing the gut bacterial quality. Daily use of this in household is making of dosha and idli, which is one of the most common breakfast among South Indians. So here we grind rice and urad dal in a definite proportion and we keep it overnight for fermentation. Bacteria will act on them and it will produce a large amount of carbon dioxide, which will make the batter very soft and fluffy. The next morning it will be ready for cooking. But in this case, actually we are giving time according to the climate. If it is warm temperature or hot climate, we have to keep only 4 to 5 hours. Within that it will ferment. Whereas if it is a cold temperature, winter season, we may have to keep up to 8 to 10 hours for fermentation. But it is done by bacteria. We don't have to add any microbe into it. It happens its own. Sometimes it overflows because of lot of carbon dioxide produced in the batter. Another domestic as well as commercial uses in the production of bread. Bread making, we need a single cellular fungus which is called a yeast. When yeast is used in baking purpose, it is called baker's yeast, and if it is used for brewing purpose, that is in beverage industry, we call it as brewer's yeast. But both are same. The scientific name of yeast is Saccharomyces cerevisiae. Once again, Saccharomyces cerevisiae, by heart it with the spelling. Now you can see bread loaf in the first picture and the second picture shows yeast. This is almost common in all the household. If you open the fridge, definitely you will find one packet of yeast. Another traditional drink of South India is toddy. It is obtained from palm tree. They make a cut at the top of the palm tree and the exudate comes out of it, that is the sap comes out of it. It is collected in a pot and it gets fermented and it becomes a very natural alcoholic beverage which is otherwise called a palm wine, that is toddy. It is having alcoholic content but comparatively less. But these are not very common in our place, but in Chinese culinary it is very important. For example, they are using fermented soya beans, bamboo shoots, fermented fish, etc. Now coming to cheese. All of us like cheese and cheese is available in different forms and different types. Among the different types of cheese, Swiss cheese is very popular. It has got large holes in it. How are these holes formed? It is by the large amount of carbon dioxide produced by a particular bacterium used for fermenting that Swiss cheese. That bacterium is called a Propionibacterium shermani. Again, Propionibacterium shermani. You have to learn these names very well. Very few specific names we are learning, like scientific names. Rest all are common names only we are telling. But if you are learning one organism, definitely study whether it is a bacterium, a fungus or a protozoan, which category it belongs to also. Another very popular and very exclusive type of cheese is Roquefort cheese. Roquefort cheese is blue in color because it is used by growing a kind of blue color mold, as you can see in the picture. As a result, the cheese also will develop blue color. It is a part of French cuisines. In this table, you can see a few types of cheese and the microorganisms used for ripening. The soft cheese is called a camembert cheese and it's Penicillium camemberti. Then semi-hard cheese is Roqueforti. It is Penicillium roqueforti. Then hard Swiss cheese is formed by Propionibacterium shermani. So in the textbook, Penicillium roqueforti is not given, but they have mentioned about Roquefort cheese. Now we are moving on to microbes in industrial products. How can we make use of microbes to produce products on a large scale? The first is in the form of beverage industry. Production on an industrial scale, it requires growing microbes in very large vessels called fermenters. Microbes are especially yeast has been used for this fermentation product from time immemorial for making wine and other beverages like beer, whiskey, brandy, gin, etc. And the yeast scientific name I already mentioned, Saccharomyces cerevisiae. It is called a brewer's yeast. Part of its anaerobic respiration or fermentation, it produces ethanol. There are different type of alcoholic beverages. It is depending on the type of substance used for fermenting, and also what kind of processing is being done, whether it is with the distillation or without distillation. So fermented beverages like first in the picture what you are seeing are wine and beer. They are formed usually without fermentation. So their alcoholic content is also less. Whereas whiskey, brandy, rum, all these are produced after distillation. As a result, their alcohol content is very high. In this picture, you can see different types of beverages with their alcoholic content. Beer is produced from barley malt and the alcohol content is 3 to 6%. So malting is a process where first we allow the barley to germinate. The germinated seeds will be allowed to dry using hot air. Then after fermenting that we get the beverage, that is 3 to 6% is only the alcoholic content, whereas wine is produced from grapes, and during Christmas season many people do it at home also. Yeast is added for this, and 10 to 20% is the alcoholic content in it. Whereas when it comes to the rest of the things, their alcoholic content is high because they are coming after distillation process. For example, brandy. It is produced by distillation of wine and alcohol content is 60 to 70%. Another useful industrial product is antibiotics. The term antibiotics was coined by Selman Waksman. This is a new term for us because we are all familiar with Alexander Fleming in connection with the production of antibiotics. What are antibiotics? They are chemical substances produced by certain microbes, but it can be used for killing or retarding the growth of other disease-causing microbes. The credit for finding out this antibiotics or human use goes to three scientists: Alexander Fleming, Ernest Chain, and Florey. If you go back to the story of this discovery of antibiotics, it's quite interesting. It was a chance discovery. Alexander Fleming, this happened in 1928. Alexander Fleming was working as a bacteriologist in one of the hospitals in London. So he went for vacation and he left some of the bacterial culture plate. If you remember, in the beginning, one of the slides I showed you how the bacterial plate is, or bacteria are cultured in agar. You must have seen the bacterial colony in those petri dishes. So here he left some bacterial growth in the petri dish unwashed, and he left for his vacation. On September 3rd, 1928, he returned to his lab and he went to the basins where he kept it. He found certain molds were growing in some of the culture plates. Mold means fungi. But to his surprise, he found that those culture plates did not allow bacterial growth. This was really surprising for him, and he concluded that certain juices produced by these molds could restrict the growth of bacteria. That means they prevented the growth of bacteria. Though he asked his team to isolate the mold and make study or what is the chemical, they couldn't do anything further. Credit for extracting the antibiotic from that mold. That mold was nothing but Penicillium notatum. Okay, Penicillium notatum, from which we first isolated the antibiotic, and it was formulated in the form of a medicine by two other scientists of Oxford University, Harvard Florey and Ernest Chain. So these three scientists were together given the Nobel Prize for the discovery of antibiotics. What are antibiotics? Actually, they are used for the control of bacterial growth or bacterial infection. The antibiotics prevent the bacteria from making its cell wall. So without a cell wall, can it survive? No. So thus the bacterial growth is prevented. So during that time, many deadly bacterial diseases were there in the society. One was plague, another one whooping cough or Kali Khansi, then diphtheria or Galgotu, and leprosy or Kushtarog. With the help of antibiotics, we could wipe out these diseases very successfully. Also, this was the discovery happened during Second World War, so many wounded soldiers could be treated using antibiotics. Moving on to the next application of microbes in using or making certain chemicals. Chemicals like organic acids, alcohols and enzymes can be produced by the help of microbes. What are they? Organic acids, alcohols and enzymes. Especially when these are naturally produced in the body of organisms, but when we need it commercially on large scale, we can use certain microbes. For example, there are three acids that we have to study here. One is citric acid, that is used as a preservative, then our flavoring agent, then acetic acid used as a preservative, and also butyric acid and lactic acid. So this is a picture shows Aspergillus niger, which is a fungus. From Aspergillus niger we get citric acid commercially. It is obtained by anaerobic fermentation of sucrose. Sucrose is a disaccharide. Acid is used to make medicine, flavoring extracts, food and candies, and ink and dye. Clostridium botulinum is the next bacterium, giving us an acid called butyric acid. Now next is Acetobacter aceti. It gives us vinegar or acetic acid. Vinegar is commercially used and it is a bacterium. So there are two stages in the fermentation process of vinegar. First step is yeast converts carbohydrate into alcohol, that is by fermentation. Second step, this alcohol that we obtain is oxidized to acetic acid by Acetobacter aceti. So in the second step we make use of this bacterium. What is the meaning of vinegar? Sour wine, and it is a French word. Humans are aware about it since hundreds of years. Vinegar is a product of microbial fermentation and it was recognized first by a scientist called Kutzing in 1837. Almost 30 years later, in 1868, Pasteur described it as biological activity. What are the uses of vinegar at home? It is used as a spice or as a cooking agent, or it is used in pickles as a preservative, used for preserving canned vegetables and fruits, and also it is helpful in curing constipation and helps in improving our digestion. Last organic acid is lactic acid, which is obtained from a bacterium called a lactobacillus. So lactobacillus, as we mentioned, it's a product of fermentation. Lactobacillus comes under lactic acid bacteria, lab bacteria, and lactic acid is a product of fermentation by Lactobacillus bulgaris and Streptococcus lactis on cornstarch, can be a substrate, or molasses from sugarcane, or potato or whey. So these two names are not given in your NCERT, you can note it down. These are the four organic acids that we studied. If you recollect again, it's very important. Take small notes on this with the name of the organism and the product that we are obtaining. So the first one was Aspergillus niger, which gives us citric acid. That was the only fungi. Rest three are bacteria. Which are they? Clostridium botulinum, which gave us butyric acid. Then Acetobacter aceti gives us acetic acid or vinegar. Third one is lactic acid bacteria, that is Lactobacillus bulgaris, which gives us lactic acid. Another commercially important product is ethanol. We talked a lot about ethanol. It is produced by the anaerobic fermentation by yeast. Third is enzymes. We know enzymes are basically proteins, so they are produced within the body. Enzymes have various functions within our body. We know, if you remember, you learned about the cleansing action of soap in smaller classes. There we learned the dirt is oily in nature. So if dirt is oily in nature, soap molecules will be helping in removing them. But these days, detergents are formulated with enzymes. Enzyme like lipase can digest oil. So if you add lipase like enzymes in the detergent, then the cleaning efficiency is very, very high. We can remove all oily stains from the cloth easily. So enzymes also need to be produced on a large scale, that is by use of microbes. And coming to bottle juices. Bottle juices are more clear than the juice that we make at home, because they use certain enzymes to clarify the juices. Which are the enzymes? Pectinases, which will digest the pectin, and proteases. As a result, bottle juices are clarified. Now we are going to study another enzyme which is used in the medical field. One is streptokinase, that is otherwise called a tissue plasminogen activator, TPA. Streptokinase is produced by bacterium called a Streptococcus and modified by genetic engineering. It's used as a clot buster for removing clots from the blood vessels of patients who have undergone myocardial infarction leading to heart attack. So if there is a blockage in the heart or in the blood vessels, usually they administer the streptokinase medicine to digest these clots. So 3 enzymes we learn: lipase in detergents, proteases and pectinases in bottle juices, and streptokinase as a medicine for removing clots. Now we are talking about other bioactive molecules, that is we obtain from microbes. Cyclosporin A is one among them. It is used as an immunosuppressive agent. We know our immune system has the function to eliminate all the substances coming from outside. They are called foreign bodies. It can be microbes, it can be dust particles or anything. But during organ transplant, this makes a problem. That is, for a person who is receiving an organ, any organ from a donor, for his immune system or for her immune system, it is a foreign agent. So what is the tendency of the immune system? Somehow reject it or get rid of it. So during organ transplant, just before the surgery, during the surgery and post-surgically, they have to take medicines called the immunosuppressive agents, which will suppress their immune system so that the chance of rejection is less. This is produced by a fungus called a Trichoderma polysporum. So these names and all please write down and learn by heart. Cyclosporin A we get from Trichoderma polysporum, which is a fungus. Another bioactive molecule that we use in medicine is statins. Statins are produced by yeast, that is also a fungus, a kind of yeast called the Monascus purpureus. We already learned another is Saccharomyces cerevisiae in baking. Here Monascus purpureus, which is used in making a bioactive molecule called a statin. What is the function of statin? Blood cholesterol lowering agent. It competitively inhibits the enzyme responsible for synthesis of cholesterol. Competitive inhibitors and all you learn in class 11. So hope you remember. It is structurally similar to the substrate, right? So here it competes with the enzyme which is responsible for making cholesterol. As a result, the cholesterol formation is reduced. So it is very good medicine for people having high cholesterol level. Hope you understood this lesson very well. Thank you for watching the video. Please like, share and subscribe to my channel, Biology My Passion.